Our next company is MaxCyte, and presenting for MaxCyte is Doug Doerfler, President and CEO.
Thank you. Thank you all very much. So the problem that we bet the company on solving is how do we increase the potency of cell-based therapies. And what we have is this is a webcam shot of the company in Gaithersburg, Maryland this morning. We have a single platform, and the platform is really to increase the efficacy of cell therapies. Over the course of the last dozen years, we've been working in a number of clinical areas to do that. We've also moved into a revenue-generating model by selling the technology into the drug discovery and drug development area, which has allowed us to be a cash flow positive company. We have a very strong commercial footprint. We're in the majority of the top pharmaceutical companies. We've either licensed the technology for cell therapy or acquired it for drug discovery and drug development. 7 of the top 7 global pharmas and 3 of the top 3 in Japan. We do this all directly. And again, our philosophy is to keep our relationships directly with these customers so we can really move this technology forward. The technology is a flow electroporation technology we originally licensed from the Center for Blood Research at Harvard. It's not like the electroporation technology you probably used in grad school. This has about 90% efficiency and 90% cell viability. It's a flow system. It's sterile, it's closed, and it's fully scalable. We can basically load any molecule, whether it be siRNA, RNA, DNA, proteins, into any cell lines, primary cells, insect cells, for instance, at any scale. We can go from very, very small scale up to 2 times 10 to the 11 cells in about 20 minutes at those kind of efficiencies and viabilities. So when you think about that, you then think about the applications, and we're going after very high value applications. We're working in the pharmaceutical drug discovery area in creating cells, engineered cells, so that big pharma can run small molecule screens against them. I'll give you an example of that in a minute. We're also working with big biotech drug developers where they want to manufacture very rapidly a gram plus of protein, monoclonal antibody, in a CHO cell line to do all their preclinical work, and there's not really an alternative to do that. So they're using our technology to do that. And third, they're using, we have our technology placed in a number of vaccine producers where they're manufacturing quick response VLP and VREE vaccines for, mostly for medical countermeasures. And then, of course, in the cell therapy development, which I'll talk about at length. This is an FDA-cleared device within CBER. The operations are ISO 9001. It's an instrument that we license or we sell, and then there's also a single-use disposable. So every time a patient's treated or every time cells are run, they use a single-use disposable, and we move on. We're the first group in the space of clinical electroporation or taking electroporation into the clinic. So we have a very, very strong intellectual property position. So in the area of screening, it's really using primary cells to do drug screens. The issue in big pharma, we talked about this a little bit this morning, was that the cells that the big pharma screeners want to use aren't the same cells that they can scale up. They would rather use more human, biologically relevant cells, but they can't figure out a way of scaling it up, and they can't figure out a way of engineering those. And that's what we're doing with them. It allows them to create new targets, more novel assays, more assays that align more with the biologically relevant, the physiological condition they're trying to eventually find drugs for. Here's an example. Sorry you can't read this, but it's an example of the work that we just did with Vertex and Flatley Labs, where in this case we took an epithelial lung cell and we were able to put in an iodide flux assay. So we took a human cell, we cultured that cell, and using our technology, we were able to put in molecules that created this flux assay so they could go after CFTR receptors for cystic fibrosis drug discovery. We did this very rapidly with them. We scaled it up. They've been publishing papers on it. And so far, they've been very, very pleased with the quality of the hits they've been able to get out of this versus the old Fisher rat cells that they were using prior. So when we talk about using technologies that in regenerative medicine for drug discovery, here's an example of that. We're also working with a number of groups who are developing IPS approaches, either loading RNA, DNA, and proteins in combination to create these IPS cells. Move past here. So let's move on to the cell therapy. We are passionate about engineering cells. We believe that the biggest threat in this field is to invest in a cell therapy product only to find out at the end of phase 3 that you have an ambiguous clinical result. So we work with companies and we work with academic centers to engineer their cells for higher potency. The approach we take is non-viral. We use no extra chemicals or proteins, a very clean system. And again, it's an automated, sterile, FDA-cleared device they can use in the early stages of academics all the way through to the patient bedside. So what we're basically doing is either taking cell lines, or in this case, autologous cells, adding in the loading material, again, it could be DNA, RNA, or protein, whatever, put it through the MaxCyte device, and out comes an engineered cell product. And you can do this in enough volume that you can actually aliquot those cells, freeze them down, and have them available for reinfusion with the patient over a prolonged period of time. So where we're really focused is being able to develop and characterize products that have a high level of biological function, the function that you're trying to align to a clinical outcome, and then have the desired characteristics so you can produce those at quantity at a manufacturing cost that makes sense, and we can all, everyone can make money off of doing this. So these are the examples of the kind of relationships that we've entered into over the last several years. On the top are companies that are using the technology to advance a number of both regenerative medicine and immunotherapy programs. And the bottom is a growing list of academic groups where we license the technology on a non-exclusive, non-commercial basis, allows them to do their clinical work, and then we create intellectual property with them that we can then move into doing other licensing deals with other big companies. And again, we're working in regenerative medicine, immunotherapy, and also in, for instance, spinal fusion. A couple examples. One is we have our colleagues here with MetaNet. We're working with them. They have a 7-site dendritic cell processing, 7 sites in Japan that process dendritic cells loaded with whole tumor lysate. They service hospitals there. It's through a very well understood regulatory path in Japan. We also have that license with them to move the technology into China, Taiwan, and Singapore. And again, we created a product for them that is a dendritic cell product loaded with whole tumor lysate that is 50 times more potent than a similar product that's done with co-incubation, which is the standard method. We also work with United Therapeutics. This is pulmonary hypertension. I'm sure you all understand this disease. It's a devastating disease, basically a disease of vascular insufficiency. The heart pumps out into the lungs, the arterioles start to degrade, and the blood pressure goes up, and eventually there's cardiac failure. So what we're doing with them is we've created a new cell drug that's an engineered epithelial precursor cell. We load it with DNA plasmid for eNOS, and we put those cells into the patient. We license them the technology, and we provide instruments, disposables, and we actually get royalties on their commercialization. These are the data set that was used in the submission for the approval for the clinical trial. On the upper right-hand side, what you may be able to see is that they induced pulmonary hypertension using monoclonal with a cohort of rats. And then after about 3 weeks, they then added the MaxCyte engineered EPCs into those rats. And you see a rather dramatic reduction in systolic pressure over the period. Well, on this side, you see 3 panels. On the left would be normal rat lungs. The center would be lungs that are representative of pulmonary hypertension. And the right-hand side would be those rat lungs that had been treated with monoclonal and then came back in with cells that were treated with the MaxCyte system with the plasmid for eNOS. And here you see a rather robust overall survival benefit. We've dosed about 10 patients in a phase 1/2. The data looks quite strong, and we're in the process right now of going into a multi-site US 2B trial for pulmonary hypertension. Lastly, talk about chimeric antigen receptors. This is a dual publication that Carl June presented from the University of Pennsylvania last summer. This is using a lentivirus to transduce T cells with chimeric antigen receptors against CD19. A very, very exciting study. He's dosed 8 patients now. I think he's got complete responses in the majority of those 8 patients. We've been working with Dr. June for a number of years, but our Our approach is not using lentivector, but actually using RNA. So we have RNA that's constructed to express a chimeric antigen receptor, has an FC receptor on the outside of the cell. It's a transmembrane anchoring mechanism, and inside the cell there's a T cell upregulator trigger, and once that FC receptor hits the antigen, it upregulates and triggers the T cell. These T cells appear to be killing 10,000 cells, 10,000 cancer cells per T cell. It's a very potent, and we're doing this again with using RNA. So it's a transient process. You can process enough cells to dose 6 times, freeze them down, and give the patient a dose every 1 or 2 weeks. The issue here is that the lentivector is basically toxic to any cell that might carry that antigen, so we wanted to do a transient system with RNA. So we're fundamentally working with Dr. June on all the solid tumors. So we have several clinical programs in place right now with mesothelioma, breast cancer. We're about to go into a CD19 trial for pediatric ALL, and then at the beginning of 2013 for ovarian cancer and pancreatic cancer. The one area that we're focused the most on and the most exciting, this comes back to our original premise as a company, is moving this away from a centralized facility and close to the patient. This is the process that Penn currently uses with the lentivirus CAR. It takes anywhere from 12 to 15 days in a large centralized facility where you have to separate and expand T cells. And with our technology, it knocks down a few days where you can actually use our technology to load the cell with RNA and you reduce some of the steps. What we're working toward is eliminating the centralized facility and actually process these cells directly from a patient apheresis. So you apherese the patient, you'll have enough effector cells in that patient, in that bag, in that leukapheresis, to load with RNA at a very, very high percentage, and then aliquot that and give that to the patient. So we believe that the future of cell therapy is not in a centralized facility, but actually is as an add-on, if you will, an augmentation to the current therapeutic apheresis markets that are currently, there's thousands of them around the world, and this is the area that we're focused our attention on, because we believe if we can get the potency up on these effector cells, it will be similar, if not better, than using selected and expanded cells. Our growth strategy really is in 3 areas. The last slide, one is direct sales expansion, so we're looking for partners to help us with our sales efforts. New applications for the technology. We come to these meetings, we always find someone that has, have you thought of this application? Love to talk to you for the next couple of days. And strategic partnerships with companies who share this view that eventually cell therapy will be patient bedside, or at least in community hospitals and not in centralized facilities. So thank you for your attention, appreciate it.